So what I just talked about are the defense system of the innate immune systems. Then I'm going to talk about the adaptive immune responses of human antiviral defenses. As you know, there are adaptive immune responses are mainly mediated by B cells and T cells. Both B cells and T cells, they have structure on the surface called B cell receptors as well as T cell receptors, BCR or TCR in short. These are receptors that can recognize and bind to foreign antigens, or so-called epitopes. That gives it really the specificity of targeting different pathogens. So each B cell and T cell, they has many identical BCR or TCR on the surface. And on the same cell, they all recognize the same epitope, but different B cells or T cells, they have different BCR or TCRs, which recognize its unique epitopes. So the epitopes recognized by BCR, they are peptide, nucleic acid, or polysaccharide, and they can be recognized recognized in solution, while the epitope recognized by TCR are mainly peptides. Here, I want to show you how this antigen recognition works. Basically, in our humans, there are cells such as macrophages, as well as so-called dendritic cells. These are phagocytic cells. They engulf, like I mentioned earlier, they engulf the microbes or viral particles, and then after the phagocytosis, the foreign microbes into peptide, they use their so-called MHC molecules on their cell surface to present the foreign peptide on the cell surface. So these are so-called antigen-presenting cells, mainly macrophage, dendritic cell, as well as B cells. Basically, the peptide is presented on the surface of antigen-presenting cells. It's sort of like raising a flag on the cell surface. And this flag really then attracts different T cells. If it is MHC class 2 molecule, the second class of MHC, you will recruit the so-called CD4 T cells, a type of T cells with it's called a CD4 protein on its surface. And then this type of T cell uses its T cell receptor basically to recognize this foreign antigen. When this recognition is formed, it really can lead to the maturation of the CD4 T cells. Basically, this recognition then activates CD4 T cells so that it can release different cytokines. Then these cytokines can actually promote the recruitment of other immunocytes, as well as promote the antibody secretion of nearby B cells. So this is really important for the humoral immunity or antibody-based mediated immunity. If the antigen-presenting cells actually has MHC class 1 molecule on its surface, which can also present peptides on its surface, and this MHC class 1 presented peptide recruited the group of T cells called CD8 T cells. And when CD8 T cells using its TCR to recognize the foreign peptide, the CD8 T cell also become activated. CD8 T cell, when it matures, it becomes the so-called cytotoxic T cells. Basically, now it evolved the ability to kill those neighboring infected cells. This is the so-called cell-mediated immunity. To summarize this part, the binding of a TCR, so summarize the last slide, the binding of T cell receptor to its epitope will cause the T cell to proliferate and differentiate into an effector cell that will be responsible for a type of immunity that can kill infected cells. Some T cells have CD4 molecule on the surface, and some have CD8 molecule on the surface. And this TCR can recognize and bind to a specific epitope if the epitope is associated with a self-MHC molecules. Here in more detail of this MHC molecule, MHC class 1, as I just mentioned, it can interact with the CD8 T cells. MHC molecules of the antigen-presenting cells will display the foreign peptide on the surface of the antigen-presenting cells, which then recruit CD8 T cells with the T cell receptor on its surface. And this interaction triggers the CD8 T cells to maturize into cytotoxic T cells, which can then become able to kill the infected cells, basically promote the apoptosis of the infected cells. MHC class 2 molecules are found on the surface of cells such as B cells and macrophages. MHC class 2 molecule can present peptide made outside of cells to CD4 T cells, or so-called T helper cells. And similarly, the CD4 T cells use its TCR to recognize the antigen peptide presented by the MHC molecule on the antigen-presenting surface of antigen-presenting cells. And then this interaction between the CD4 T cells then triggers the CD4 T cell to release cytokines into its environment. And then this, if the antigen-presenting cell is a B cell, then this cytokine can then reactivate the B cell and promote it to proliferate and differentiate into so-called plasma cells, which are the main type of cells secreting specific antibody. In addition, it also can help promote the B cells to differentiate into a memory B cell, which can then can last for a long time. That's the basis of our immune response, the memory of our immune response. Then the antibody, as you probably know, then is a powerful tool. When it's released by the B cell, it can directly neutralize viruses by coating the viral particle with a protein shield to make the virus innate no longer able to bind to its host cell receptor. In addition, they can also recruit other mediators such as phagocytic cells as well as complement molecules, basically to indirectly inactivate the virus. So in summary, this part I talk about the innate response and adaptive response to viral infection. The innate response can through phagocytosis or through interferon system or through the NK cell mediated cell killing. The adaptive response mainly can be characterized as T cell responses as well as B cell responses. Keep in mind the B cell responses is also dependent on the T cell responses, mainly the CD4 mediated B cell activation.